Okay, once I set those up as so, like I said, does it matter what order I have those in? If your order is different than mine, is that okay? Yeah, we should still have the same ratio. Now, as I look at this, my only one for this one was y little r when I FOILed it. Did I need to put all of those? No. If I have the same thing, I don't need to do it 4 times. That's just making busy work. I only need to do it the 1 time. So basically, when I have this 16 square, I'm not going to use this part of it. I'm only going to use those top 4 squares. So when I do my cross, it's going to be capital Y, lowercase y, capital R, lowercase r. Capital Y, lowercase y, lowercase r, lowercase r. Lowercase y, capital R, lowercase r. And finally, y, both lowercase, r, both lowercase. Now, when we talk about a genotypic ratio, what are genotypes, folks? What is a genotype? Huh? The actual alleles, meaning what? The actual alleles, meaning what? The letters, right? So, if I'm doing a genotypic ratio, I look and I say, okay, I just start with this box right here. I say, what is the genotype? It is capital Y, lowercase y, capital capital R, lowercase r, right? Now, are any of those other boxes that genotype? No. Okay, so what is this? 1 out of 4. Now, like I told Dylan the other day, what I'll do now is I'll put a semicolon to separate. What's my next one? Capital Y, lowercase y, lowercase r, lowercase r. Are any of those like that one? So what is my ratio? 1 out of 4. My next one is lowercase y. Let me leave a little space so I can put my lowercase y, capital R, lowercase r. Are any of those the same? No. So that is 1/4. And finally, my last one is going to be 1/4 lowercase y's and lowercase r's. So my genotypic ratio is just actually writing out what the letters are. Yeah, go ahead, Maddie. If I did the whole thing, it would have been 4 out of 16. Okay, which reduces to be what? 1/4. Yeah. Okay, so if I would have filled out the whole thing, it would have been 4 out of 16 instead of 1/4. Now, what is a phenotype? Brandon? The physical appearance. So let's look at the very first one right here. Okay? Right here. What is the physical appearance of that? It is heterozygous for both. Okay, so what is it? Yellow and round, right? Are any of the other ones yellow and round? No, so what's my ratio? 1/4. What's my second one here? Well, it's also yellow, right? But it is yellow wrinkled, right? 1/4. Do I have another one? Absolutely. It is green and round. Do you understand how we got green round? The lowercase y is green. It's homozygous recessive, and that's green. Capital R, little r is heterozygous, so it's going to show the dominant, which is round. What is my last one then? It is homozygous for both recessives, so it's 1/4 green and wrinkled. Whoa. Right? Are we okay with that? Because we're going to work a couple of more. Let's open our interactive notebook. Open your interactive notebook to page, I believe it's 26. That sound right? I believe it's 26. So, ladies and gentlemen, this is the first one. This is our first example on page 26, right? Well, it might not. Yeah, 26 is the directions, right? No, this is on 27, right? This is our first example on INB, page 27. In pea plants, the round seed allele is dominant over the wrinkled. The yellow seed allele is dominant over the green. Isn't that what we were just dealing with? Okay, so those are the same. Round, wrinkled, yellow, green. So it says, a plant that is heterozygous for seed texture and seed color is crossed with a wrinkled and heterozygous for color. So let's deal with the first one. Right here, we're going to put, on these two lines here, we're going to put the two parents. And the first one is heterozygous. So, what is my genotype of my first parent? Big Y, little y? Big R, little r, correct? My second parent is wrinkled and heterozygous for color. So, what is my phenotype? Or excuse me, my genotype? The Y's come first, so my color comes first. It is heterozygous, so that is what? Capital Y, little y. And it is wrinkled. To be wrinkled, it has to be both lowercase r's, right? Now, based on what I showed you earlier, okay, based on what I showed you yesterday, 2 times 2 equals, this one's going to have 4 gametes, right? I'm going to have to FOIL that one, okay? What is this one going to have? 2 and 1, how many? 2 gametes, right? Do I need to FOIL that one? Can we see what the 2 gametes are? So, as I bring this down to my crosses here, I am going to FOIL my red ones. So my first, okay, then my FO, my outside, then my inside, and then my last, So I have now done FOIL to get my 4 gametes there. How many gametes do I have for my other one? 2. What are they going to be? Big Y, what else? Little r, and little y, little r. Can you see that? Do you understand? Dylan, you understand how we got that? Olivia? Understand how we got that? What about back there, Miguel? You okay with how we got that? All right. Now, I don't need these boxes down here, right? So I can just cross those out because I'm not going to use these boxes. When I go through and make my cross, capital Y, capital Y, capital R, lowercase r. Capital Y, lowercase y, capital R, lowercase r. Capital Y, lowercase r, lowercase r. Capital Y, lowercase y, capital R, or excuse me, lowercase r, lowercase r. Capital Y, lowercase y, capital R, lowercase r. Lowercase y, capital R, lowercase r. Capital Y, lowercase y, lowercase r, lowercase r. Finally, lowercase y, lowercase y, lowercase r, lowercase r. So I've done my cross, correct? I've done my cross now. How many total boxes do I have filled? 8. So when I do my ratio, it's going to be however many out of 8, right? However many out of 8. So as I come down here, okay, as I come down here, these are my possible genotypes. Excuse me, check. These are my possible phenotypes, correct? So as I'm going through there, and looking, I can make little tally marks, okay? Round yellow is the first one I'm doing, okay? If it is round and yellow, what's it going to be? It's going to have a big Y, right? And a big R, both of them. Does it matter whether it's homozygous or heterozygous? No. So I count. Here is a big Y, big R. 1, 2, 3. I have 3, don't I? So I come back down here. 3 what? 3 out of 8 are round yellow. How many are wrinkled yellow? So I'm looking for a capital Y and lowercase r's, right? Here's the capital Y, lowercase r. 1, 2, 3, right? So what's my ratio? 3/8. How many round green do I have? Okay. Round green is going to be what? Little y, little y with a capital R, right? How many do I have? 1 out of 8. How many wrinkled green? 1 out of 8. These numbers, 3, 6, 7, 8 should equal up to 8, right? 8 out of 8. Okay? Now, my next one. Okay? This big, long guy here. In humans, there's a disease called phenylketonuria, PKU, which is caused by a recessive allele. People with this allele have a defective enzyme and cannot break down the amino acid phenylalanine. This disease can result in mental retardation or death. Let capital E represent the normal enzyme, so lowercase e would represent the PKU enzyme. Also, in humans, a condition called galactose intolerance or galactosemia, which is also caused by a recessive allele. Let G represent the normal allele for galactose digestion. Okay, so lowercase g is the, how do I want to say, the recessive allele is the one that does not digest, okay, galactose. If two adults were both heterozygous for both traits, what are the chances that a child is going to be completely normal, that the child has PKU, that the child has galactosemia, or that the child has both diseases? Okay? So, you're going to be doing a dihybrid cross of two heterozygous, EEGG with EEGG, right? So once again, can you FOIL that? First, EG. Outside, E lowercase g. Inside, lowercase e g. And last, lowercase e lowercase g. Now what about the other side? Same thing, right? Because they're both heterozygous. So if I did the first one right, I can just fill in the second one. Go ahead and make your cross. So I'll make mine up here. Okay. I've made my cross just like you should have made your cross, correct? Now, as I come down and look, it says, what are the chances of having a child that is completely normal? What would be a genotype of that? it would have both dominants, right? So as I go through, I'm going to check in red ones that have both dominants. That's it. How many do I have? How many red checks? 1, 2, 3, 4, 5, 6, 7, 8, 9. So what are the chances of a completely not normal child? 9 out of 16. The second question, okay, is that it has PKU. In order to have PKU, you need to have what? 2 little e's. Well, I'm going to go through in black and check ones that have 2 little e's. Oh, it said only, didn't it? Okay, just. Okay, so that one's out of here. Okay, I'm going to have to redo this one. So that's out of there. I checked that one. That's one that has both of them. Okay, it had 2 little e's, but it also has 2 little g's. So only PKU would be how many? 3 out of 16. How many have just glycosema, which is going to be ones that have lowercase g's, right? Yep. 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 So how many have only glycosemia? 3 out of 16. And finally, how many have both diseases? How many have both? 1 out of 16. Right? Are we okay with that? Can you do this, folks? Because we're going to be doing a lot of it in the next few days.